Third lecture.
Roman numeral 4C. And then we'll go to histology. I want to give Glenn his histology lecture. It's far more than I can do in a single day. And I have some eye clickers right at the beginning. These eye clickers are just survey questions to see if the system works. So I just downloaded. I have a new base and new software. So anyway, we'll just be testing the system today. So let's start out with that. So if you want to go ahead and turn on your devices, and if you push the on button twice, as you probably know, it changes frequency. So just push it once, and if the battery's working, somebody told me their battery wasn't working, it should light up blue, and then if the response is received, it should light up green. I guess the new ones even give you more information than that. Okay, so just some fun questions to start it out today. What year are you in college? So let me go ahead and start it. I usually base this on 30 seconds to see how that goes as the semester progresses. I can always increase it. Okay, let's see how it came out. Most of you are seniors. That's what has been true in the past. However, there are a significant amount of graduate students, and most of you are in your last year. Yippee. Second question. What program you're in? This is kind of interesting for a number of us, so let's see what that comes out to be. Okay. Almost all of you are getting a BS degree in biology, 92%. By the way, I want to meet with the people in the Masters of Physics, Medical Physics program, if you can, just for a few minutes after lecture day, if you want to meet me in the front of the room, just to discuss a couple things if you're here today. Okay, question 3. What do you want to do with your life? I should have put undecided. Don't know. Okay. Yeah, 90% of you, that's usually the case, which I used to think was ridiculous, but we're doing very well in that area now. Okay, question 4. It's useful to me as well as interesting to everybody else to know how much physiology experience you've had, so it helps me actually in planning this class. Okay. It's actually faster than the opium it is. That's good. 30, about a third of you did have the 2085, 2086, and a third of you had high school courses. A few had the ZO46. That's the wrong number, but the comparative morphology and comparative comparative physiology class. Some of you had both, quite a few. Anyway, it's kind of interesting. Only a third of you had no experience, so almost no as a third each of the other classes, I guess. And finally, number 5. It's always interesting to find out what kind of grief you go through and how bad of a mood you're in when you get here. Whoops, that's not what I meant to do. Oh, did I stop? No, sir. Wow, that's horrible. Wait till you see this. 13%. 22 of you drive greater than 80 miles to get here. Boy, I thought I had problems. That's horrible. Over an hour, hour and a half to get here. That's insane. I'm sorry. Anyway, yeah, there's some sacrifices going on. That makes me even want to do better. Okay. So let's go on then. I'll download it all. If your data works under your program, under the Blackboard gradebook, I'll put some numbers up, which I'll erase after today. But just to show you that you're being received, I'll put it up on Blackboard, maybe 5 points or something. But then I'll erase it because we'll start officially on Thursday. Okay, as I said last time, the volume of cells is definitely a reflection of the solute concentration that they're in. And we use the term tonicity to express that effect. In other words, the tonicity, whether it's iso, hyper, or hypo, is a way to describe the effect of a solution or a solvent on cell volume. For example, since cells are 300 milliosmol, if a cell is placed into solution that's 300 milliosmol, the cell, much fluid will enter as will exit, or there's no net fluid movement. And so we call that solution, that the cell's in isotonic solution, which is what we want to do if we're going to have an IV, for example, or keep a tissue in a physiological solution. If the concentration is higher than that, we would expect water to leave the cell, going down its concentration gradient, and the cell to decrease in volume, and we call that a hypertonic solution. And the cell shrinks, we refer to the term, as far as cells is concerned, as crenation. And finally, if the concentration is less than 300 millios, the cell accepts more water. In other words, there's water would move into the cell down the concentration gradient or diffuse in, and that causes swelling of the cell, and if sufficiently low, it would cause lysis, or the cell to burst. Okay, so that's about where we quit last time. I have this slide in just to show that the cells that are easiest to study with isolated cell preparations are blood. So to grab a graduate student, it used to happen to me when I was a graduate student, they'll take your blood out to use for some biological experiment. So anyway, most of these early studies were done on graduate students' blood. But if you put a blood in a hypertonic solution, it looks like little kind of prickly or little protuberances coming out from the cell like this. If it's isotonic, the red cell has a biconcave disc shape, which is the largest surface area per unit volume structure. We'll talk about that when we get to the respiratory system. And a hypotonic, it swells. Very hypotonic, it lyses. And then you end up with what we call red cell ghosts. And if you look in the solution, they literally look like little balloons with nothing in them. And we're going to look at that preparation in lab this week. We're going to do that type of activity. The terms of osmolality and tonicity are not synonymous. And where they differentiate is what we call the reflection of a given solute for the membrane. if the solute is lipid soluble, which in classic transport terminology means it's not reflected to the membrane. In other words, if you're measuring the solute concentration of something that's permeable to the membrane, and some examples are glycerol or urea, those statements, hypotonicity and isotonicity, are not synonymous. I got knocked twice. I wasn't trying to overemphasize. I don't know what happened. So anyway, 3 milliosmolar solution of a glycerol or urea solution would be hypotonic. And the reason is that these solutes go right into the cell. So if you have 200 milliosmolar urea, the urea will go down its concentration into the cell, increasing the solute concentration within the cell. And then water will follow urea, and then more urea will come in and more water, and more urea and more water, and the cell will get larger and larger and larger. So the reason is that the urea is permeable through the membrane of the cell. The same is true of glycerol. So any solute you have that's significantly permeable through the cell, even though it has the same concentration, for example, of solute within the cell, can be hypotonic and can cause lysis. And that is a biologically important phenomenon, not awfully important, but it is. It's something that you should be aware of. So tonicity and osmolarity are only synonymous if the solute you're discussing or measuring is reflected or impermeable across the membrane. And most ionic solutes, such as sodium chloride and so forth, are effectively impermeable to the membrane, and so in that case the terms are synonymous. Membrane translocation, the movement of substrate across the barrier, occurs by many different mechanisms, and what I will do now is talk about some of the mechanisms that substances go across the membrane. Very few important biological molecules can go right through the bilayer. It can occur with some small hydrophobic substances, but anything hydrophilic, it's almost impossible to go through.
Sir, can you try to adjust your microphone?
Oh, is it not working? I probably bumped it in my, yep, looks like I turned it off. Nope. Hello? Maybe the mic, looks like the battery quit. It's working, but just barely. I'll turn on this one and try and stay in this location, which I'm very poor at. Okay, so anyway, significant amounts of material can only move through bilayers by means of protein mechanisms. And the first and simplest to consider is a pore, which is simply a column, a cylinder would be a better description, a cylinder of protein that goes across the matrix of a bilayer. And one example of a significant pore is the ones we use for water translocation called aquaporins, of which there's a family of many different types, and we'll get into that later. But pores are used even when you have small molecular types, in most cases, to get solute across the membrane. Another mechanism of getting substances across the membrane is what we classify as channel. And the difference between a channel and a pore is channels are pores with gates or with doors, if you will. So the only difference between these two is this has a mechanism to control passage of materials through it. So we call it gated. And what opens and closes the gates are a variety of different things, and we'll be talking about this off and on through both semesters. One of the most common is voltage. There's a voltage difference across membranes, and we're going to define that in detail in a week or two. But if you change that voltage, many of these gated channels open or close. And so one mechanism is changing the voltage on the membrane. Another is ligand, and I used that word last time, but a ligand is a molecule that binds to a specific receptor on the membrane, and some of these gates open and close when bound to a specific ligand. And finally, another family of these gates are dependent on second messenger interaction.
and that's when the ligand binds to another receptor. There's a series or cascade of events that occur in the cell, and one of the byproducts of those events is something that opens and closes the gate. And we'll talk about all types of those type controlled channels through the class off and on. But so another way that substances get through membranes is through these gated channels. And another characteristic of them, and also for the most part for pores, is that they're selective. They're selective for a variety of substances, sometimes extremely selective. For example, they can definitely detect differences between cations, such as sodium, potassium, calcium. All these move through very selective cation channels that are gated. So we'll talk about opening and closing those channels and what that does to cells through the class. And finally, so those are two ways that solute gets across membranes, through pores and channels. A third is through very specific carriers. And carriers are molecules that couple to a substrate or a ligand, if you want, on one side of the membrane, and then are taken by action of that carrier to the other side of the membrane. So they have specific binding sites for one or more solutes, and then they translocate that solute to the other side. And here's a very simple model of a carrier. So X is a molecule that binds to one of these carrier molecules, a specific binding site that's open. In this case, on the, looks like this is the inside of the membrane, not that it makes any difference, inside of the cell. And then once bound, the carrier molecule goes through what we call an allosteric transition, or changes its molecular state, and exposes that binding site to the opposite side of the membrane, and then the X comes off, or the solute comes off. So it keeps going through changes in conformation, and when it does so, it moves solute from one side to another. Many of these carriers, which we classify as either active or passive, are termed facilitated diffusers. In other words, diffusion is a passive process where substances move down their concentration gradients, but if it requires one of these carriers to mediate that diffusion, we call it facilitated diffusion. Another family won't be translocated without the imposition of some energy source, usually hydrolysis of ATP. So there's both passive and active translocators, and we'll give you examples of a few today and many throughout the course. I put this in to make a very serious point, and that is that the carrier molecules can't turn themselves within the membrane. That would be blocked by the amount of energy that would be required. They'd be energetically limited or impossible. In other words, what has to happen is a change in what we call a conformation of that protein, so that one side, one way it's conformed, the active side is exposed to one side of the membrane and the other conformation to the other side. And this is a very simple model. They called it the ping pong model, where the solute being transposed, hooked on one side, in this case outside of the cell, and then there's change in conformation, and then it's released on the opposite side. And that is possible and does occur. The idea that you could turn the whole protein in the matrix of the bilayer is impossible. There are several very classic characteristics of these carrier systems, whether they're active or passive. And we always start out this discussion with some of these really important characteristics of any type of carrier-mediated translocation process. And the first is it's saturable. In other words, it may be above physiological levels of the given substrate, but it can be saturated. And many of these, that plays a role in health. I'll give you some examples when we get to the kidney of that particular situation. So this graph represents, just as a way of picturing this, the concentration of the substance on the abscissa of the graph and the rate of diffusion on the organ. So this would be mass per unit time, and this would be millimoles or milliequivalents or whatever. So with an increase in the concentration gradient across the barrier, that's what the x-axis would be showing, there'd be an increase in diffusion. And the blue line would be what we were talking about with Fick's equation last time. It's simply the result of diffusion being primarily mediated by a difference in concentration. So the rate of diffusion is directly proportional to the concentration gradient across the barrier. So that's simply Fick's law. But substances that move through a carrier don't work by Fick's law. And this is many times how we found out carriers had to exist. And that is exemplified by there's a Vmax, and you increase the concentration of the solute on one side of the membrane, and then it would approach asymptotically that Vmax and wouldn't exceed it. And that's simply because you've saturated all the available carriers. It's a very simple thing to picture. If you were working on an assembly line and your job was, one summer I worked on an assembly line and I was helping put together first aid kits, and my job was to throw in a little tube, I think it was Vaseline or something, when they went by. And if they went by too fast, every so often one of the kits didn't have my little tube of Vaseline in it. So it's typical of working on an assembly line. It's limited by how fast the carriers can facilitate the process. It's the same thing. If you have the substance moving too fast by whatever membrane you're talking about, and there's not enough carriers, you're going to reach what we call a Vmax. And there are many situations biologically where that occurs. And as I said before, some of them are very important clinically. Another characteristic of carrier-mediated transport is its specificity. And in this, what we're indicating here is that each carrier, that active site or binding site, is specific to very specific or moderately specific to substances. And usually they're in groups. For example, the amino acid translocators work in families of amino acids, like basic amino acids or amino acids. There are specific families of amino acids that are transported by specific carriers. But it certainly wouldn't be possible for amino acid transporter to carry glucose across the membrane. So there are specificities, and the specificities are usually in groups closely related structurally molecules. Specificity is not perfect. Often within carriers there's competition, which leads right to the next little story. And for example, if you had glucose and galactose both carried across a membrane by the same carrier, you could get this situation. So this is a little bit hard to try and show you by a graph. But let's say you're measuring how much glucose gets inside the cell per unit time, and so you have a set concentration gradient in this case, but you're just measuring how much gets there per unit time. And the rate slows down because the concentration gradient disappears with time. If you add galactose at the same time, and they're both transported by the same carrier, the galactose and glucose will compete for the same carrier, and so it'll cut down the glucose translocation. And there's a lot of examples of where that's useful and biologically important. So there are many examples of competition between carrier molecules that has implications biologically. And finally, not only is there saturation, specificity, competition, but there's also something which we call non-competitive inhibition. There are many carriers that we can poison various ways, and one of the classic ways is place a molecule in the pocket, in the binding pocket, which gets stuck there but won't be carried. And there's many examples of that. But this is a way of blocking translocators, or these carriers. And one example of that is glucose and fluorescein. So this is the same glucose kind of kinetics that I showed you on the previous graph. So if you're measuring how much glucose it goes into a cell dropped into a glucose solution by itself, you get A. But if you added fluorescein before you put in glucose, then the rate of glucose would not near be as fast. because some of the carriers are bound up to fluorescein, and so there's few left that carry glucose. And you can easily differentiate between non-competitive and competitive inhibitors, and this is a little bit harder to understand. But if you add more glucose and increase the glucose gradient in this non-competitive inhibition, it won't change the kinetics at all. In other words, let's say we double the concentration gradient, it's not going to affect this slope at all because you have very few translocators available for glucose binding. But if you go back to this situation where you're looking at the competition between glucose and galactose, if you increase the concentration of glucose, you'll increase the likelihood that glucose will go into the cell, so this line will go up. By the way, this graph, I think some of you will probably ask this question later. Let me make sure that I'm mentioning this graph correctly or describing it correctly. When I said the first instance, you're looking at glucose only in the solvent, and the second, you're looking at glucose and galactose in the solvent, but we're only measuring glucose arrival inside cells. And so this is an A and B getting into the cell. This is glucose getting in the cell in the presence of both galactose and glucose. So you inhibit how much glucose per unit time gets into the cell in that situation. If you added more glucose, though, it would be more effective in competing with galactose, and so this line would go higher. And that was my little point here, that won't occur with non-competitive. No matter what the A concentration is, you won't get any higher slope for this second line. So you can kinetically differentiate between non-competitive and competitive inhibitors. Another characteristic of carrier transport is very rarely is it a uniporter, we call it. Almost every biologically important molecular carrier molecule is capable of hooking to more than one substrate at a time. In other words, we call a uniporter, exemplified here, a carrier molecule that hooks only one solute. Some of these, if the higher concentration is on the one side, it'll take it in. If it's on the other side, it'll take it out. So some of these can move substrates either way, but it only hooks to one molecule. There are many, many molecular carriers that are coupled, and that usually occurs because the binding of one of the two solutes changes the binding site of the other, makes the other more available. And so they require two simultaneous solvents to couple to them before the allosteric transformation will occur, before the molecules are available to the other side. So those are coupled or co-transporters, and some of these co-transporters are symporters, meaning they simultaneously transport those two molecules to the other side of the membrane, and some of these coupled transporters are called antiporters because they transport substances in the opposite direction. Let's see, I guess I've said all this. Some examples of these are given in Boren Bullpape's book. Don't memorize every example. Just wanted to give you some stories, and by the time, if you survive with this all 2 semesters, by the time the semester's up, you'll be able to come up with these because we talk about them so often in the class. But many, many organic translocators couple the sodium simultaneously. So in order to get glucose into most cells, there's a type of facilitated diffusion in which sodium simultaneously couples to the carrier at the same time. And so that's sodium glucose co-transporter. We call it SGLT, sodium glucose transporter, and it's very prolific in most cells. There also are, this is again another organic solute, sodium amino acid co-transporters. There's a sodium and bicarb that takes the bicarb out of cells. And here's one that brings phosphate in. So many examples of these symporters in a variety of cell systems. Antiporters are also prolific. This is a sodium-calcium antiporter that, when sodium goes in, takes 3 sodiums to pull 1 calcium out. This is a sodium-hydrogen. One of the principal ways we get hydrogen out of the cell is with this exchanger, we call it. And then there's even ones that simultaneously hook to 3 different subs at the same time, with sodium going into the cell with bicarbonate pulls chloride out. There's almost every configuration you can imagine. Some of them become very important as far as understanding processes, and some are not. Often, one of the solutes moving down its concentration gradient gives the energy for the other to come out. And we're going to talk about that. Many of those are called secondary active, but I'll give you a better description of that in just a second. And so, we've been talking about carrier-mediated transport and all the characteristics of it. I said some of it's passive and active. If it's just simple facilitated diffusion, we're usually talking about a uniporter that takes the substance one way or another. But some of these passive carrier-mediated processes are coupled to 2 substrates or more, and they're called symporters or antiporters. But now we'll go into another area, and that is the need for some carriers, and relatively few, to take solute across the membrane requiring metabolic energy. And this is a hopefully quite specific definition of that. So active transport in which a carrier is required. So it has a lot of the other characteristics of carrier-mediated transport. That is, it is specific, it can be inhibited, and so forth. But it usually occurs against the gradient, and not only potential gradient or gradient in charge, but sometimes also a chemical gradient, and it requires metabolic energy to expend in order for that process to occur. By far the most common and discussed of all active transporters is sodium-potassium ATPase. We'll be talking about it many, many times through the course. And this, present on all biological cells, exports 3 sodiums out of cells in exchange for 2 potassiums. It requires ATP, so here's a very simple depiction of it. So this is outside the cell, this is inside, so sodium is pumped out of the cell in exchange for potassium, and it takes the hydrolysis of ATP to ADP to facilitate that process. And indeed, that's a good fraction of the energy cells require is to keep that pump going. And secondary mechanisms is responsible for a lot of the other activity of cells that we're going to talk about, such as generation of potentials and on and on and on. There's a very specific inhibitor of this, Wabane, and we'll refer to it off and on. But Wabane specifically blocks this pump, and you can study the pump and its effects by use of what we call this cardiac glycoside, we call it, because of the effects it has on the heart, which are interesting. We'll talk about that later too. But it poisons this pump. Okay. So here's a detailed picture of this pump's activity from Bourne and Voltape's book. And so we'll start out here with step 1. And it, in essence, is an enzyme that's stuck on the membrane, but it goes all the way through the membrane, and so it's available to both sides. So it's essentially a carrier, but it won't change its configuration or conformation until there's imposition of energy. So when we begin it, we'll be at the, we'll say the step 1, the binding sites for sodium are exposed to the inside of the cell, and at that point, ATP binds to the thing. This is before. Then that allows the that is one. So that allows sodium then to be bound. So that exposes the ATP binding exposes the binding site for sodium on the inside of the cell. Sodium binds to its binding sites within the protein, and then that binding causes the conformation change which allows sodium to be exposed to the outside. And then with the next step is the hydrolysis. Actually, the hydrolysis, this is wrong, this diagram's correct. The hydrolysis of the ATP is what causes that occurs right here. That's what causes the conformation change. So the hydrolysis causes conformation change and keeps it cycling around. Oh, here, I'll get it right here. Where's the hydrolysis? Here's phosphate, phosphate, phosphate. I guess they show it occurring right here. So the hydrolysis causes the site to go from this conformation to that. But anyway, ATP molecule binds and causes this shift, or going up, exposing this site and allowing sodium to go through. So anyway, the diagram's correct and goes through step by step what happens to the molecule as the hydrolysis and binding takes place. In the presence of wabane that binds, as they show right here, to the outside of the membrane, that the conformation blocks the pore and it becomes non-functional. So you see the pore where the potassium binds is blocked, and so potassium can't bind, and then the pump stops. So it stops it in this particular conformation right here. There are another family of, quite a family of ATPases that translocate other substances. Two of the most important I'll mention are calcium ATPase. It's not prolific, but it is in sarcoplasmic reticulum and other membranes of our body, and actively translocates calcium out of cells and requires directly, the pump itself requires calcium. And then in distal nephron cells in the kidney and many other places, there are specific pumps that pump hydrogen, like in the intestinal, in the stomach. And this hydrogen or proton pump many times can create large gradients of hydrogen across membranes. So anyway, those are two very important active transport carriers for calcium and hydrogen. Another thing I wanted to mention in this introduction to different transport systems is secondary active transport. And there are examples of both symporters and antiporters that are called secondary active. The carrier itself does not require energy, but it depends on energy at another carrier to occur. And the best example that I've ever seen is sodium-calcium exchanger. And so in this example right here, you have a very large gradient in sodium-potassium in all cells because of this is sodium-potassium ATPase. So in all biological cells, sodium is pumped out in exchange for potassium pumping in. So that leaves sodium just a few millimoles inside and 120 millimoles or so outside. So you have this large gradient of sodium, and potassium has the opposite gradient. Well, what happens is this carrier, this sodium-calcium antiporter, uses the sodium gradient to move calcium out of the cell. So the carrier doesn't work unless both sodium and calcium couple to it, and so it uses the diffusion gradient for sodium going back into the cell to pull calcium out. So there's no direct energy link itself. I put this little wiggly line which represents motors in schematics to say that this requires ATP. This pump does not require ATP, but it creates a concentration gradient. So here we have a passive pump that creates a concentration gradient of calcium, and it's a huge one, about 100-fold difference in calcium inside and outside cells, in many cases only due to this passive pump. So it uses the sodium diffusion to create the calcium concentration gradient, and a very important biological active pump. If you poison sodium-potassium ATPase with wabane, the calcium concentration will go up within the cells, which proves that it's secondary. It requires sodium-potassium ATPase for this pump to work. Okay, so that is the last slide of that lecture, and I would like to introduce the next lecture, at least get somewhat into it. So this is lecture 3 in your outline. So now we're going to go into histology and then skeletal, muscle, and so forth. So for the next quite a few lectures, we're going to move back into anatomy, and these lectures will come out of the anatomy book. And this particular one's, I think, the second or third lecture. But there is one small part on connective systems of cells that's only in Borum and Volpe, very nice section in the book, but most of this comes out of histology. Out of anatomy. Okay, histology is a term which we use for the study of tissues, and tissues are cells of a body that have the same function. The anatomy book has a very nice section in the beginning of this chapter that talks about our bodies are like big, complex manufacturing companies, and we have a division that does this, and accounting, and a division that does receiving, and so forth. And our bodies are like this. We have tissues that perform different tasks, and the cells in those tissues are very specifically differentiated to perform those tasks. So we're going to talk about those different families of cells and their common microscopic structures, and then look at where they're located and so forth. There are 4 very general classes of tissues in animals, and the first is called epithelia, and these are tissues that have on one side of the tissue either the outside of the organism or some body chamber. So they're on surfaces, not only the skin, the outer surface of our body, but the surface, the mucosal lining of the intestinal tract and of the bladder and so forth. So the apical side, we call it, of the cells of epithelia opens into a chamber on the basilar side of the basal membrane. So they line ducts and cavities and surfaces of our body. Connective tissue is a very diverse family of tissue. They bind, support, and protect. For example, the skeleton is a connective tissue, but so is the blood, and so is adipose tissue. So we're going to talk about these probably mostly in the next lecture, but it's very diverse and very interesting class of tissues. The third is quite similar in that they all have the ability to contract or to shorten. We usually think first when we're talking about muscle, skeletal muscle, moving our skeletal system, our muscle, our limbs around so we can walk and manipulate ourselves in the environment. But there's also muscles involved with bladders and with the heart that produce pressure. So we're going to talk about those muscles in the next week or two and how they work and their histological characteristics. Nervous tissue, finally, we'll talk about mostly the last half of the first semester. Very distinct family of tissues that we use primarily for communication between the cells of a multicellular organization system, very quickly called acute control, and regulation of our system. We'll go into that mostly in the second part of the class. Before we get into the different families of tissues, wanted to look at those few pages in the Borum book that tell how cells are coupled together and what we know about it. And it does apply to many of the subjects we'll discuss during the next couple of months. Points of contact between cells are called junctions, and there's many different classes of these junctions. The first one we'll talk about are referred to as tight junctions, and they're represented on this figure right up here at the top. This is an epithelia that forms a boundary between, for example, the lumen of the intestinal tract and the basal membrane below the mucosal. So this would be the lumen above the cell. We call this the apical side of the cell, and this down here referred to as the basolateral side of the cell. Well, the borderline between the apical and the basolateral is called the tight junction. So these wiggly lines are connective systems between the cells at that tight junction site. And as you can see here, and on the next figure, I guess it's not shown so well on the next figure, so let me back up. It's composed of rows of little proteins that couple the two cell families together. And you'll notice, first of all, that it's at the apical border. It's right at the place where the luminal fluid could come into contact with the fluid in the interstitial compartment. And it's called a fluid tight compartment because that's the blockage site of material going between the lumen and the interstitial fluid. And to prove that, if you use some electron-dense material and take a photograph, this is horseradish peroxidase, you'll see if it's in the lumen, it can't get past that border, or if it's in interstitial fluid, it can't get past that border. Some people say it's like the O-rings on a cylinder that won't allow things between the walls of the piston and the cylinder, but it's a very tight, fluid-tight junction between cells. And not only that, as I said in that lecture on the fluidity of membranes, that also blocks proteins that are part of the membrane of the apical side from getting to the basolateral side, vice versa. So it also results in there being membrane domains or very specific proteins that are on the apical border of cells and on the basolateral side of the cell. This is a close-up of the architecture, a drawing of what we think this tight junction looks like. And it's made of rows of proteins called claudins and occludins. And these are proteins shown, they're multipass proteins that are part of the matrix of a membrane, but they have high affinity to themselves, and so they bind to similar proteins in a neighboring cell and form because there are many rows, they form a very tight junction. There is differences between how many rows of these proteins there are, and membranes that we call tight membranes have many rows, and those that aren't have fewer rows. So some epithelium really restrict passage between the cells, and some are not so successful. They're not a strong, rigid attachment. They just attach by charge differences, but it's enough to restrict flow between the cells. What is the tight junctions, or mechanically hold cells together, are anchoring junctions instead of just a lipid tight junction. So anchoring junctions are in two families. The first family I've listed are desmosomes, and this is representative of the desmosome. And cytologists like to refer to it as a spot weld between cells. There are little spots, as represented here, that attach one cell to another, and they depend on two molecular species to couple them together. One is the intermediate filaments, part of the cytoskeleton within the cell. So these intermediate filaments anchor plaque, they call it, or these little spots on the different positions on the cell wall. And those plaque deposits are the anchoring site for adhesion proteins called cadherins. So cadherins exist in these little, they're shown here green, between the two cells, and they take a complement of cadherins from one cell, that couples to cadherins from the other cells, and hooks the two cells together. They call them cadherins because they're calcium-dependent binding molecules. They depend on calcium to change the conformation, so these molecules have a very high affinity for each other. And one of the ways which we separate cells when we're doing tissue work is by removing all the calcium from the interstitial spaces or between the solute surrounding cells. And if so, these linkages just fall apart. So they're calcium-dependent junctional proteins. Here's some dimers representing cadherins. So they exist as dimers coming out from both cells, and then in presence of calcium, they have very tight affinities for each other. So cadherins work both with the previous junction that I was showing right here, with the desmosomes. These are cadherins here, but cadherins also occur with the other family. These are the desmosomes, with the other family of anchoring junctions, which are called adherens junctions. And adherens junctions are slightly different than desmosomes in that they exist in just below the tight junctions, just below the apical border of cells, in belts, just about like the belts that tight junctions exist in, but they're coupled inside the cell to another type of cytoskeleton element called actin, which, as you may remember, is part of muscle physiology. It plays a role has a high elasticity. And so this type of junction is one in which the cell can change its shape and they can still be coupled. And they're thought to play a big role in tissues during differentiation as the tissues reach their final shape, as well as tissues that undergo stretch or distortion in their lifespan, because a lot of elasticity is possible around these adhesion belts, they call them. Both desmosomes and adherence junctions depend on cadherins as their anchoring molecule. And finally, the last junction between cells has a lot of functional role in physiology, and we'll refer to it in detail when we get to the hard and to smooth muscle and so forth, and that are gap junctions that are referred to here. Not all cells are interconnected by gap junctions, but they're very important in some cells, and these are essentially cylinders that go right between cells, so it allows what we call cytoplasmic continuity. They're essentially an open channel between two cells, so the cytoplasm of the two cells is continuous. And also, electrically, they're low resistance pathways, so whatever occurs electrically in the membrane of one of the cells occurs in parallel to the other cell. And so signaling processes occur both as far as membrane potential events and even signaling molecules. Intracellular second messengers can go through these gap junctions. So they enable cell signaling, and they'll play a role in all types of interesting activity biologically. This is the molecular structure of these gap junctions. On each membrane, they consist of a group of transmembrane proteins called connexons that cluster together in an array of 6, forming a column. And then two of these in two cells, two adjacent cells, come head to toe, if you will, or in register, allowing continuity between the two cells. And so the group of 6 connexons is called a connexon, and then two of these together forms a channel between the two cells. We'll talk about those off and on. They are important in cardiovascular physiology, especially in how hearts are electrically continuous. They work in smooth muscle throughout our body and other tissues. So we'll save that story and their function when we get to those areas. Okay, so now after that little aside as far as how cells are connective systems, we'll talk about first epithelia, and hopefully get through some of that today, and then starting next time we'll go through the other 3 classes of tissues, starting with the connective system. But functions of epithelial tissue, one is protection, physical protection, because of course the skin is a epithelia, and so the skin protects us from changes in temperature and osmolarity and all types of immune function and so forth. Also, around all cells, it controls the permeability and has a great effect on cell size. We've been talking about many of our sensation devices, such as touch and temperature and pain, are relying on sensors that are in the skin. And finally, we'll talk about glands that are present in epithelial tissue. In fact, glands are part of the epithelial family, and the epithelia talking about different types of glands. And so we'll first talk about membranous epithelial tissue, and then the other would be glandular. And as far as the first subdivision, you can differentiate membranous epithelial tissue as either simple or stratified. And the simple epithelia is one that has a single cell layer thick, or stratified just like strata on the side of a mountain, or layered cell clusters or layers of cells. And we'll see both in this depiction. So we'll start talking about simple epithelia. And the second characteristic is all epithelia have an apical border, which I've already referred to, which is open to a chamber or to the the inside mucosal side of whatever tissue you're looking at, and then a basilar side that hooks the basal membrane tissues underneath. As a rule, epithelia do not have blood vessels going through them. They're non-vascularized, and the connective tissue below them is where the blood vessels are located. And the epithelia are the family of tissues that are most mitotically active or constantly growing. And hence, when you block mitoses, for example, in things like cancer treatment, you slough or lose the lining of your digestive system and so forth. So it's a very mitotically active family of cells. The first group we'll talk about of simple epithelia are squamous, and they're one cell layer thick, but these cells are very flat. They almost look like a pancake with a berry in between. The book says they look like pieces of a puzzle, if you will. But they're very thin cells and irregularly shaped, and they're adapted. Their function is to allow substances to filter across them and diffuse them. They're located in places where you want to allow fluid movement across their barriers and so forth. And so some of the logical places are the alveoli lining alveoli, or the airbags within the lungs. The inner layer of all blood vessels is called endothelium, and it's made up of this simple squamous tissue. And another classic capillary bed that we look for endothelium is the glomerulus, but these are capillaries are always a likely place to see simple squamous epithelium. As you get thicker, it's still simple epithelium, but now the cells are cube-shaped, and we call them cleverly cuboidal cells, with a nucleus occupying the center portion, and this is typical of a cell in the kidney nephron or tubule. And these function, so these cells would have more organelles, so they would have more capacity for such things as secretion or production of a fluid that goes into a lumen, or reabsorption, absorption, or secretion. So these are where cells have the intracellular organelles to allow, cellular activity. So they do far more than filter. They start secreting and absorbing and so forth. Glands and the pancreatic cells, we'll talk about them and where these cells are located and their function in those locations. Columnar cells, moving along that same thought, would have even more organelle complement. They're tall, column-shaped. Usually the base of membrane is closer to the basilar side of the cell. They function, again, in the same family of functions, secretion, excretion, absorption. But here we have even more organelles allowing even more activity. So they would be found logically in the lining of the intestinal tract and the colon, where lots and lots of absorption and secretion are going on. Some of these have specialized cells that look like goblets that produce lots of fluid. They're called unicellular glands, and I have a better figure of that in the gland portion coming up. The goblet cells are especially adapted columnar cells that just produce a very copious secretion, and I'll show you an example of that shortly. Some of these columnar epithelium are lined with cilia, such as those of the uterine tube that facilitates the gametes going down the uterine tube. Some of them have microvilli that just increase their surface area. So these would be ciliated columnar epithelium. And some columnar epithelium are called pseudostratified and also ciliated. And they're called pseudostratified because it looks like they're several layers thick. But all of them attach to the basement membrane. Not all of them go all the way to the top. So we really can't call them stratified because there's not layers of cells. All the cells contact the basement membrane. So these are called pseudo, as in fake stratified, and this type of epithelium is always ciliated. And so they're a unique family. They're only present one place, and that is in the respiratory system. They line the trachea of our system as well as bronchioles. The cilia help in removal of the particulates we inhale, but we'll talk about that in more detail when we get to the respiratory system. And finally, moving on to stratified classes of epithelium. Stratified squamous is found on the surface of our body, our skin, and there it's keratinized, which simply means the outer surfaces of the skin, the cytoplasm is just changed slowly into a protein matrix called keratin, and we'll talk about that in detail in just a couple days when I get to this. However, there are also stratified squamous. It doesn't have these outer surface cells containing keratin, and so it's called non-keratinized. And those are present in orifices that open out into the body, out of the body, such as the anus, the vagina, the oral cavity. And so it's, again, squamous cells that form layers. Usually the most basilar cell is more cuboidal than squamous, but they classify them as stratified squamous cells, and they're not keratinized. Stratified cuboidal cells are a fairly rare family. There's only a couple places they're formed, and that's where every layer is specifically cuboidal. And the primary place they're found is within glands, like the sweat and the salivary glands, so only in the tubules of those glands, and very rarely you see a couple of tubules with double layers of cells, so it's a fairly rare class of epithelium. Transitional epithelium is not stratified nor simple. It's halfway in between, so they call it transitional. And the reason is the cell layers essentially slide past each other. They must not have very strong desmosomes. Maybe it's mostly adherence junction. But these cells have the ability to go from a very thick epithelium to a thin epithelium, and the difference is the bladder's full here and empty here. So as you stretch this epithelium, it has the ability to cover a larger surface area, and so essentially you're going from a stratified in a way to a simple epithelium. However, it looks like it's almost exactly like the pseudostratified, except it's not ciliated, nor does it have a lot of other characteristics. But anyway, we call it transitional. About the only place you find it's in bladders. So I put in a table from another text that shows the characteristics of all these epithelia and where they're found and some of their unique functions. It kind of puts the whole thing together. And let me just introduce glands, and then I'll let you go. Glands are a classification of epithelia, but they produce substances and secrete them. And they're classified a lot of different ways. One is by whether or not they have ducts. And we're going to talk about this second semester. But endocrine glands are glands that don't have any ducts. So the gland does not specifically put its product out into a specific location, but secretes it into the blood that goes to the whole body. Where an exocrine gland, such as the pancreas or the salivary glands, have ducts that allow their product just to go to a specific site. And finally, you can differentiate glands as to their cellular organization. And this is where I was going to talk about the goblet cells. But these are the goblet cells of the digestive system. And they are part of columnar epithelium, but they only produce a product. They're not there to absorb or secrete. They produce a product that's secreted right into the lumen. They're called goblet cells, and they're found in digestive epithelium primarily. I think that's a good place to go because we got another big subject. So we'll start here on Thursday.